you. So without further ado, our first speaker is Dr. Beatriz Mejía-Mercado. She's a research data analyst, quantitative ecologist at the Florida State University Coastal and Marine Laboratory, or FSU-CML, where she oversees the seawater monitoring system. So this is a real-time water quality monitoring that has multiple stations in the northeastern Gulf of Mexico. She received her PhD in oceanography from Florida State University, where she used underwater imagery to study deep fish community on seamounts on one of the world's largest marine protected area, the Papahana-Umokuakea Marine National Monument in Hawaii. So in addition to her work at FSU-CML, she continues to conduct research in deep sea ecology with special interest for spatial patterns, environmental influences on deep sea communities, and the association between fish and habitat-forming species. And today, she will present some of that work to us.
Okay, great. Well, thank you so much for all these people. I'm so happy to see how many people are connected today. Thank you to Maeva and Lucy for organizing this seminar and allow me to be the first participant. So today I want to share how my work across continental basins and Pacific seamount systems has gradually changed the way I think about deep sea communities. The deep sea is often described as one of the most uniform environments on Earth, dark, cold, and seemingly homogeneous. But when we actually observe these systems, we see something very different. Communities that are highly structured across space. So the question that motivates this work is, where does that structure come from, and at what spatial scale does it emerge? This is a compilation of all the work that I have been doing in the deep sea. And so I'm going to start first in the Gulf of California, where broad geological and environmental gradients shape communities at large spatial scales. From there, I'm going to move to seamount systems in the Northwestern Hawaiian Islands, where local environmental variability becomes increasingly important. And I will briefly step back to ask a methodological question. How much of what we observe depends on how we sample these systems? Finally, I'm going to zoom into a final scale habitat associations where corals and other habitat-forming organisms influence local community structure. By the end, I'm going to put all these different scales together into a broader hierarchical framework for understanding how deep sea biodiversity is organized across space. So before moving to the individual systems, I want to step back and focus on the broader ecological context connecting this work. The deep sea is characterized by conditions like darkness, high pressure, and limited food availability. And under those conditions, we might expect biological communities to be relatively sparse or randomly distributed. But observations from many deep sea systems suggest that these communities are not entirely random. Species often appear associated with particular depths, habitats, and environmental conditions. So this raises an important ecological question. What processes structure deep sea assemblages across different spatial scales? And so to show you that, or to answer these questions, I'm going to show you how environmental gradients, geomorphology, and habitat variability may contribute to those patterns. And so why is this important? Because understanding whether communities are structured is important not only ecologically, but also practically. And also, if biodiversity is organized predictably across space, then our monitoring system needs to capture that structure accurately. However, in the deep sea, that becomes especially challenging because what we observe is already filtered by the technologies and sampling approaches that we use. We are talking about different platforms, sampling designs, and observational methods that can all influence the communities we observe and how we interpret ecological patterns. And that directly affects biodiversity assessments and conservation decisions, including vulnerable marine ecosystem identification. So if deep sea communities are structured across space, the next question becomes, what environmental processes are generating that structure? One of the most common variables used in deep sea ecology is depth. But depth itself is not a direct ecological driver, because when we talk about depth, we are talking about multiple environmental changes occurring together. As depth increases, temperature decreases, oxygen often declines, pressure increases, and water masses shift. So changes across depth actually reflect multiple interacting environmental gradients operating simultaneously. And to explore how those gradients shape communities, I will now take you through two different or contrasting ocean systems. The first one is the Gulf of California, a continental basin environment characterized by relatively continuous environmental gradients. In these settings, communities experience smoother transitions across large spatial scales. The second system is the Northwestern Hawaiian Islands, where communities occur on isolated seamount systems separated by large distances. In these environments are much more heterogeneous, and the local environmental conditions can vary substantially even at similar depths. So as we move from continental basins into seamount systems, the ecological context becomes increasingly localized and especially complex, creating what many now describe as deep sea seascapes. And to investigate how these different spatial systems shape ecological organizations, I use different complementary vehicles, imaging vehicles, including AUVs, autonomous underwater vehicles, ROVs, remotely operated vehicles, and HOVs, human-occupied vehicles. And I use these tools to examine how community structure emerges across different scales. I'm going to start in the Gulf of California, a tectonically active system in the Eastern Tropical Pacific. This region provides an ideal setting for examining deep sea communities across broad environmental gradients. Geologically, the Gulf formed through the separation of the Pacific and North American plates. And over millions of years, tectonic activity created a series of deep basins connected by transformed faults, producing a highly complex seafloor landscape. To study this system, we use observation collected in 26 dives using the ROV JSON aboard the research vessel Atlantis. These dives cover depth ranges around 1,600 meters over 2,300 meters and provided continuous visual observations across multiple basins. Together, all these dives give us a regional perspective on how deep sea communities were distributed across contrasting basin environments and geological settings. Here, we have three examples that represent different basin environments and geomorphological settings that we surveyed during that expedition. These bathymetric maps highlight steep slopes, basin margins, crater-like structures, and transformed fault structures that create a highly heterogeneous deep sea landscape. At this scale, bathymetry and tectonic structure generate strong environmental gradients across the seafloor. The red transects that you can see here are the paths that were conducted by the ROV JSON from deeper depths to shallower in each structure. So across this surveys, we observed a wide diversity of deep sea fauna, including crustaceans, cephalopods, fishes, echinoderms, sponges, and other benthic organisms. But some of these organisms also appear associated with particular depth zones and seafloor structures, suggesting that basin morphology may influence habitat distribution across the deep seafloor. Together, these observations highlight how geological complexity can contribute to the environmental variability that supports diverse deep sea communities throughout the Gulf of California. But geology is not only part of the story. Once the physical structure of the basin is established, environmental gradients operating within those basins start shaping how communities are distributed. One of the strongest gradients in the Gulf of California is oxygen availability. Oxygen becomes especially important because it's one of the major physiological constraints in the deep sea. In this system, oxygen concentrations change dramatically with depth and across regions of the Gulf, creating an extensive oxygen minimum zone. This oxygen minimum zone contains an oxygen concentration less or equal to 1 milliliter per liter. Here, we can see how in green, the southern Gulf contains a shallower and much more intense oxygen minimum zone compared to the northern part of the Gulf in red. And so to examine how these conditions influence communities, we analyzed video transects collected from 7 dives in the Gulf of California from depths from about 350 to 1,300 meters. Under such extreme low oxygen conditions, we might expect biodiversity and ecological organization to collapse, but we documented thousands of organisms representing a wide range of deep-sea taxa across these oxygen gradients. Here we are looking at how community composition changed across oxygen gradients in the 7 dives that we analyzed. Oxygen concentrations ranges from extremely low conditions below about 0.2 milliliters per liter to relatively higher oxygen environments approaching 0.7 milliliters per liter. This is still within the oxygen minimum zone. The color areas represent the proportional contribution of major faunal groups within the assemblage. And here you can see the major groups associated with a specific color. So what we see is that multiple major faunal groups remain present even under extremely low oxygen conditions, but the relative abundance changes across the gradient. So rather than collapsing into random assemblages under low oxygen stress, these communities remain highly structured across the gradient. Oxygen acts as a strong environmental filter while still supporting organized ecological patterns at the basin scale. And that raises the next question. If a strong environmental stress can still produce organized communities at the basin scale, what happens when we move into systems where local environmental variability becomes even stronger? Seamounts systems provide an ideal setting to explore that question, because unlike continental basins, seamounts are highly heterogeneous environments where conditions can vary substantially across different sites of the same seamount, even at comparable depths. We are talking about ocean currents, substrate composition, and seafloor topography that can all change over relatively small spatial scales within these systems. And we found that with this work that was focused on Necker Island in the Northwestern Hawaiian Islands within the Papahanaumokuakea Marine National Sanctuary that before was Marine National Monument. We use here AUV surveys conducting between 200 and 700 meters to examine how these local environmental differences influence deep-sea fish assemblages within the system. What emerged was a much stronger influence of local scale variability than that we observed in the Gulf of California. And so at this point, the story begins to shift from broad basal scale gradients toward fine scale environmental heterogeneity that increasingly structures seamount communities. So in this MDS ordination, each of the points represents a single AUV transect collected in 3 different sites of the seamount, the northeast, the west, and the southeast on Necker Island. The colors and symbols indicate the different depths that were sampled across the seamount. Points that cluster more closely together represent assemblages that were more similar in composition. So here we can see 3 main clusters. These are the species, the more representative species for each one. And we have shallower depths, intermediate, and deep depth. And so if depth were the dominant driver of assemblage structure in this system, we would expect transects from similar depths to cluster together. But instead, although some depth-related clustering are present, as we can see, there is still a substantial overlap among depth groups. So much of the variation appears associated with the spatial location around the seamount rather than depth alone. And what we found was that different sites of the seamount experience different environmental conditions, including variation in currents and substrate characteristics. So even at similar depths, assemblages can remain especially distinct across the seamount. We are seeing that ecological organization is still present, but now it emerges at different spatial scale than what we observe in the Gulf of California. And importantly, this pattern was not unique to Necker Island. To test whether these spatial patterns extended beyond a single seamount system, we examined also another seamount, Pioneer Bank. This seamount is still within the Northwestern Hawaiian Islands. So in this MDS ordination, each point represents a single AUV transect, and the color symbols here indicate the different sides, the three different sides of the seamount, northwest, south, and northeast. Two broader depth-related grouping emerge here. We have a depth from 300 to 450 meters, and then a deeper one, 600 meters. However, even within those depth ranges, fish assemblages remain especially distinct across different sides of the seamount. This became very important because it suggested that the patterns observed at Necker Island were no isolated site-specific anomalies. Instead, similar spatial organization emerged across multiple seamount systems. This observation suggests that fine-scale spatial heterogeneity may be a broader ecological characteristic of the deep sea seamount ecosystems. So while broad environmental gradients still contribute to community composition, ecological organization increasingly reflects local spatial variability within the seamount environment. And so after observing these patterns at individual seamounts, we expanded the analysis across multiple systems within the Northwestern Hawaiian Islands. So the question now became whether these spatial patterns remain consistent across the broader region. And so to examine that, we conducted AUV surveys to another seamount, French Frigate Shoals, and we standardized depths, 300, 450, and 600 meters depth. And sampling multiple sites of each seamount allow us to compare communities under similar depth condition across the three different seamounts. So then we use multivariate analysis, as we used before, to examine how assemblage structure vary both within and among the three seamount systems. And so here, each panel represents each of the depth, 300 meters, 450, and 600 meters, and each point represents a single transect. The gray scales is showing the three different seamounts. We have circles, gray circles, Necker, triangles, French Frigate Shoals, and asterisks Pioneer. And so points that cluster more closely together represent assemblages that were more similar in compositions. If depth alone were structuring these assemblages, we would expect communities sampled at the same depth to become more similar across seamounts. But instead, we are observing substantial spatial differentiation among seamounts and among different sites within each seamount. So even under comparable depth conditions, assemblages remain especially distinct. Across all three seamounts, a consistent regional pattern emerges. Community organization reflects a strong horizontal spatial variability within the seamount environment. When we look across all these systems, a broader ecological pattern begins to emerge. No single environmental variable fully explains deep sea community structure on its own. Instead, ecological organization reflects multiple interacting processes operating across different spatial scales. In the Gulf of California, broad ecological and environmental gradients shape communities at the basin scale. Across the seamount systems, finer scale spatial variability becomes increasingly important. So habitat complexity, substrate variability, oxygen conditions, and local environmental context all contribute to the patterns that we observe. What becomes important is that the dominant ecological signal changes with spatial scale. So broad scale gradients define the environmental framework, while local processes refine the communities that ultimately emerge. In other words, deep sea ecosystems appear hierarchically structured across nested spatial scales. Together, these observations align closely with emerging deep sea seascape ecology. But if these systems are hierarchically structured, an important question still remains. How much of that structure is influenced by the way we observe the deep sea itself? And so this another layer allow us to find this answer. The ecological patterns that we observe are real, but the way we sample the deep sea can influence how those patterns are detected and interpreted. So here we compare fish assemblages survey using two different observational vehicles, AUVs and HOVs. The analyses were conducted at comparable depths on two different sites of Pioneer Bank. And in the MTS here, each of the points represent a single transect, while the colors and symbols indicate the different sites that were sampled, northeast in green and in blue south. So what became apparent was that some assemblages differences were also associated with survey vehicle, even when sampling similar environments. In our analysis, vehicle type accounted for roughly 20 to 20% of the observed variation in community structure. So it's important to notice that this doesn't erase the ecological organization that we observed earlier. Instead, it shows that observation methods add an additional layer to how biodiversity patterns are represented in imagery-based datasets. With different vehicles, we observe the seafloor differently because they vary in altitude, viewing angle, lighting, navigation, and image resolution. And those differences influence which organisms are detected and how assemblage structure is ultimately characterized. So rather than replacing the ecological signal, observation influences how strongly different components of that signal are resolved in imagery-based datasets. And so if we zoom even further, ecological organizations begins to emerge at the scale of individual habitats. Here, you are looking at example of deep sea fishes associated with habitat-forming corals. These images illustrate how organisms are often closely linked to the specific structural features of the seafloor. Corals and other habitat-forming organisms create localized areas of structural complexity within the deep sea. Fish communities are frequently observed in close association with this habitat, likely reflecting the importance of shelter, feeding opportunities, and local environmental conditions. So at this scale, ecological patterns begin to reflect direct habitat associations rather than only broad environmental gradients. So across the talk, we move from basin scale geological structure to local environmental variability, and finally to fine scale habitat associations within individual communities. When all of these observations are considered together, a clear hierarchical pattern begins to emerge. At the broader scale, geological settings define the physical framework of the system. Within the framework, environmental gradients such as depth and oxygen conditions influence which assemblages can occur. At the individual scale, we have seamount systems, horizontal spatial variability further modifies those patterns. And then at the finest scales, habitat-forming organisms contribute to localized habitat associations and community structure. And even observations itself become part of this hierarchy because the way we sample these systems influence the ecological patterns we detect. So deep sea assemblages are therefore not random collection of species. Instead, they reflect nested ecological processes operating across multiple interacting spatial scales. And so why is this important? Because if deep sea communities are structured by interacting processes across multiple spatial scales, then this framework has direct implications for how we study and manage these systems. First, it improves how we interpret biodiversity patterns because ecological observations only make sense within their spatial environmental context. Second, it supports more accurate identification of vulnerable marine ecosystems, or VMAs, because ecological importance is determined not only by which species are present, but also by how communities are distributed across habitat and environmental gradients. Finally, it improves imagery-based ecological assessments by recognizing that the way we observe the deep sea influences the patterns that we detect. That becomes especially important in deep sea environments where imagery is often the primary tool available for research, monitoring, and conservation. So understanding how ecological structures emerges across scales can directly inform biodiversity assessments, monitoring strategies, conservation planning, and management decisions. And this becomes especially important when imagery is used to identify VMEs. Our recent work has shown that without standardized imagery-based approaches, the same ecological patterns can be interpreted differently across regions and management systems. Different indicator taxa, density thresholds, and classification criteria can all influence whether an area is recognized as a VME or not. So as a result, similar imagery dataset may lead to very different conservation and management decisions. And the goal of this work was to help move toward a more consistent and standardized approach for VME identification, one that considers not only a species presence, but also a spatial organization, habitat associations, environmental context, and observation effects. So improving consistency in interpretation ultimately leads to stronger biodiversity assessments and more effective deep sea conservation. So where does this work go next? One important direction is predictive modeling. If we understand how deep sea communities are structured across spatial scales, we can begin forecasting how assemblages may respond to environmental change and disturbance. Another direction is expanding these analyses beyond individual systems. The long-term goal is to move toward broader comparative approaches that allow us to evaluate biodiversity patterns across regions and ocean basins. There is also a strong opportunity to further integrate ecology with conservation and management, translating these insights into practical tools for monitoring and conservation planning will become increasingly important as deep sea ecosystems face growing environmental pressures. And ultimately, this work is about improving our ability to understand, interpret, and protect deep sea biodiversity. So I hope that the main message that you can take today from my talk is that deep sea communities are not random distributed. They are shaped by interacting processes operating across multiple spatial scales, from basin scale geology to environmental gradients to local habitat complexity, and even the way we observe these systems, ecological organization emerges at every level. Understanding those patterns is essential for improving ecological interpretation, monitoring, and conservation of deep sea ecosystems. Thank you.
Thank you, Beatriz. I think you really highlighted how, the deep sea is just not one ecosystem. It's a very complex, melting pot of different pieces of the puzzle.
Yes.
We can open the floor to question. I think we have a good 10 minutes for the question. Is there anyone who want to speak out? Otherwise, I can see some questions coming up in the chat as well.
No question.
Okay, so I can read out one of the questions that is in the chat. Art from LK said, thank you very much for your wonderful presentation today. To start off, I was highly impressed by the high quality graphics in your slide and couldn't help but wonder how they were made. Did you utilize AI tools? How do you work with do you work with a professional designer?
I use AI tools. Yeah. I think that we now need to go deeper in these tools that we have. So yeah. Thank you so much. Yeah.
So speaking about AI, it's a little bit hot topic these days. Do you see an efficient way to use AI towards those goals of integrating all these different hierarchical scales and, figuring out pattern on or better, I guess, make predictions?
Well, I can say that AI are tools, right, that we have there. And in the same way that when internet started and we were looking for everything in internet, you need to choose what is it's good, in what things you can trust, uh, what kind of information is important or not, or so yeah, I mean, tools help you to make things easier, better, and faster. But obviously, yeah, you need to discern what kind of things are good or not, or how you can use these tools better.
Are there any good tools for identifying species from
Oh
video
yeah.
imagery yet? Yes.
Well, we are working right now in all these tools that allow us to, put the photo there and identify immediately different species, or not species, like groups of sponges, corals. With fishes is more, or has been more difficult, right? Because we need to include I don't know, many images from one specific species because in the way they swim, so you can see different sides of the fish, and sometimes, it's blurry, it's no, but yeah, I mean, we are using right now that when you spend like many hours in front of videos before, now you can spend much less amount of time analyzing or identifying videos with these tools. So yeah, I mean, and there are many of them free there that people are working on improving, and yeah.
See?
You are muted.
Sorry, thank you. Um, I was actually wondering because you mentioned that you saw different results based on the different methods that you used. I was wondering if you could go into a bit of detail about like what how the communities differed between the two kind of methods that you used in your transects.
Yeah. So in the first analysis, we use just one tool, AUVs, and then we, I start wondering. So there are differences? Are there differences between these tools? And so we add the HOV imagery from Pioneer Bank. And what we found is that even that we were trying to standardize the two tools, right? And so to reduce the number of images in which the HOV was going closer to the objective or things like that. but still there are some differences. So HOVs, they can go closer to the substrate or caves. And so we have rockfish. Rockfish, usually you find them, in caves or holes below the rocks or near. And so the AUV can go at a specific speed, right? And just take photos from the top. Whereas HOVs can go in an angle that allow you to see all these cryptic species that are hiding under the rocks. So this is, for example, with rockfish, this is one of the differences. It was easier to see these species from this group with the HOV instead of AUV.
Yeah,
So
that's great. Thank you. I wasn't familiar with HOVs, so yeah, that's actually really interesting to know. Thank you.
yeah, sure.
And we do have a couple of questions in the chat. Shall I read them out, Maeva, or do you want to? Yeah, so Monica mentioned that you have a very impressive number of transects and was wondering how long were they, and do you use any tools to help annotate the large amount of images that you got?
This is a very funny thing. So all the images that I work with, I annotated by hand in Excel. So in the old-fashioned way. It's been like 8, 10 years ago. So yeah, I didn't use any AI tool or no, it was using just Excel and looking at the image and annotated everything. So yeah, right now I am trying to implement these AI tools to allow me to identify organisms. So if I need fishes, at least it's gonna tell me these images have fishes. You don't need to look, to all of them. So this is the fun part right now.
Thank you. And then there's one more question from William. William says, thank you for the very interesting talk. Could you talk a bit more about the challenges surrounding species identification? Maybe some tags with important gaps. They state that we're facing a growing shortage of taxonomists and often have the impression that robust taxonomy is not fully acknowledged as the starting point of many studies that seek to inform conservation efforts. So yeah, a question about the challenges of species identification.
Well, I say again, with fish, it's very challenging because using images, you cannot see all the characteristics that we know we need to identify them until species level. So I want to say here that in our studies, we were very conservative and we give some specific numbers of taxonomy certainty. So if I was very, very sure, it will be number one if to get the species or to go to the species level or genus level. But then if I thought is this a species, but I am not very sure, I'm gonna leave it in the family level. So I give another number. So this is the first part to consider. So challenges, light. So light, sometimes we have a specific species described with a red color, but the light didn't give us this red color that we needed. So it's very challenging with images. The other part is sometimes it's blurry because the fish is swimming. And so you can see a part of the body, but not the whole body to be able to see the shape. What else? Sometimes you need to count the spines, right, to identify, for example, rockfish. And you don't have this beautiful image that you can zoom in, with all this high resolution that you want. And so you are not able to go specifically to the species level. What other challenges? See the fins, right? Dorsal fin that you need to see sometimes to make the identification. But But still, I think that we can do a lot of things with imagery, and we are using imagery for deep sea. So each time we are improving more and more the way to do it, and we have more information out here, so it's easier over time. Thank you.
Thank you, Beatriz.
There's another interesting question in the chat that asks, how would one go about learning more on the topic? You know, if like an artist or, you know, just non-scientists. Because I think the dissemination is often what keeps some stereotypes in people's mind, you know, that the deep sea is uniform and things like that. So what would you recommend to go and dig deeper?
To go and dig deeper, but to learn about this specific topic or the in general?
Community structure in the deep sea.
Well, I would say that there is still a lot of information. There is a lot of information out there. If you want to learn more, go to specific papers, you know, to have to be sure that you are knowing from people who has the experience and expertise on the topic. We recently were talking about in a workshop that we were that we need to have this beautiful movie for everybody, you know, to be Hollywood or to present about the deep sea and everything that we are just talking here. Because, you know, there are movies a lot about everything, but not about the beautifulness of the deep sea and all these interesting things. But yeah, there are a lot of books, very good books, important, I mean, very well written and with a lot of pictures. I don't know, probably be connected to the Deep Sea Biology Society and the seminars and other societies too. Yeah, I think this is the way to learn more and read papers or, yeah.
That's right. The Deep Sea Biology Society website is a good starting point. We have a Discord channel as well now, where it's very easy to reach out to the community. And maybe we can have one more question.
Sorry, there are also cruises that are extreme in real time, and we have access to that. So you can look. I don't know. I don't know if right now we have any happening, but you can always look in the website and yeah.
That's true. I wonder if we provide links for this or if we aggregate those things on our website. That could be something we could do.
It will
in the
be a
future.
good idea.
Yeah.
Yeah.
I think that would be cool. Yeah, I think you've gone around all of the questions. So join me in thanking Beatriz for her presentation.
Thank you.
Thank you for having us. And I did learn a lot.
Happy to hear that.
Yes, thank you so much, Beatriz.
We're going to take now a 10-minute break, and we will see each other a few minutes before 1 p.m. UTC time. So stay tuned.